Hi, everybody. It's Kim Curry here, and I'm bringing you the next steps of the MyGene project. I am going to skip over the finite details that I have covered in previous videos. So if you are finding that you're a little lost on some specifics, then I recommend maybe having a quick view of the previous videos created. So I'm here on the full report of my example of the YAP1 transcription factor that I've used a few times so far. And what we're going to do today is to dive a little deeper into the protein, the coding region of your gene. So clicking on your gene again in this graphic view, you'll see the mRNA sequence, which we examined last time. And this time we're going to look at the protein sequence. So going to the graphical view of this one, you can see that we're looking at the protein database here. You can get the Gen PEP records. This is the same as the GenBank record, except it's for the amino acid sequence. And you can see this graphical view as well. Now, this example, the YAP1 from the yeast, is quite a straightforward one, but it has a really neat feature that I want to show you in this example. So what you're looking for today are any of the features that get listed here. So in this example, there's only this one region feature, and it's a region feature. It says CDD. CDD stands for Conserved Domain Database. And basically what that means is that there are known domains. So in this example, the basic leucine zipper domain, this is a known domain that's found in many other proteins. It's also found in the same protein from many different organisms. And based on the fact that the sequences are the nucleotide and amino acid, the translated sequence is similar to itself, it usually means it's a highly conserved and an important functional region of the protein. So what you're going to look for this week are any functional features that you can find out and what they are. So in this example, if I hover over this one, you'll see there's a CDD link. And if we go to that CDD link, we're going to find out a little bit more about this domain. And when you come to this page, this is going to include more general information. Not only will it include information on other organisms, but it will also on the family. So you'll see that there's a lot of talk in this description here about YAP subfamilies and other related and similar sequences, like the YAP4 and YAP6, for example, in the description. So what I want you to do is to just have a look at this general description here and determine what functional details you can pull out of this description. Obviously, the explanation of some of the other genes is not what we're looking for. And in this example in particular, it starts to dive into those details of what this basic leucine zipper domain does. And so essentially, in this region, it's a dimerization location. When these two locations are dimerized, then they will bind to DNA. So that makes sense. This is the transcription factor. Transcription factors tend to bind to DNA or other proteins that are bound to DNA. But in this example, it has this DNA binding site. Sometimes you can find these features specifically called out here, but not always for all domains. So going back to the protein graphical view, you'll see here there's also one additional region, another domain here. So we can go into that domain and have a look at it as well. This one, the PAP1 domain. And so this region is cysteine rich, and alkalization of cysteine residues following treatment can mask the accessibility of the nuclear exporter CRM1. So these are details that will be helpful when you're trying to look up your gene and protein and find out what function that protein plays in the cell. So these are little hints about this. this feature in this YAP1 in particular. So the other thing about this particular example is that it has a protein 3D structure. Now, not every single protein in the database is going to have a 3D structure, so don't worry if yours does not. And if we click on the image and open this page, it will bring you to a larger image of the 3D structure. And if you click on 3D view, it will load this page. It will spin on its own, or you can actually move your mouse to spin it around and examine it. So you get an idea of what the structure of a portion of this protein looks like. So it's not always necessarily the entire protein structure that you're looking at. Might just be, as is in this example, a region of that protein. But it's still very, very neat to be able to look at it in this view. So you might be able to capture a little video or an image of this, and you can certainly use that if it's available for your protein for your research collection. Okay, one other example I want to take you to. So if you're good, you're welcome to carry on. I am going to just show you one more example using the GRM6. This one is a little bit more complex. So this is the protein graphical view for GRM6. And what you will notice is there's a lot more features on this page. We have the mature peptide amino acid features, and we have a signal peptide feature. So what this is telling me, just on first glance, is that there is actually a portion of this there is a portion of the original protein that gets cleaved. This signal portion gets cleaved off, and the resulting mature protein is just a little bit smaller. So have a look and see if there are any examples of that for yours. Certainly, the next step would be to look into what this is, what the signal peptide feature is, and there'll be links to the records in here so you can dive a little deeper and find out what that's all about. And then you'll find that there are region features with this CDD. So these are going to be back to that conserved domain database. These are going to be large known domains. And so there's a couple here. There's the periplasmic binding protein. There's also the ANF receptor. So you're going to want to go into all of the domain or region features that are present for your protein of interest. And another really interesting thing here are some region features. Here, there appears to be a glutamate binding site. So you can take note of that, where it's located on the protein. And then there are these site features. So there's glycosylation sites right here. And then there are these transmembrane regions that are shown as well. And what's interesting is that these transmembrane regions, there's 7 of them in total, they actually line up, not surprisingly, with this general CDD domain that's been identified called the 7-transmembrane receptor. So these areas here actually break down the regions of the alpha helices that would traverse the membrane. So have a look at your protein and figure out what conserved domains are present and potentially what kinds of functions they serve and what that might tell us about the protein itself. You will probably have noticed that in this example, there is no 3D structural information. And so this is, for this particular example, is not an option for this sequence. And that might be the case for you as well. That's no problem, of course. So keep a view of this one, do that printer-friendly PDF again, and hang on to the GenPep record as well as the FASTA record, so you've got the quick links. You can always pop back to them. And so, yeah, that's your goal for this next week with the investigating the protein a little bit further.